And I turn it over now to Paul Huber, who is a professor in the College of Science and has a fantastic poster on the use of xenophos oocytes for testing toxicity of nanoparticles. So check it out.
Good morning, and as Kathleen just mentioned, this morning's session is on scientific considerations. So there'll be a focus on academic research on nanomaterials, but I think there'll be a special emphasis on nanotoxicity during these sessions. Our first speaker is David Grainger, who is a George S. and Dolores Eccles Presidential Endowed Chair of Pharmaceutics and Pharmaceutical Chemistry. He's also a professor of bioengineering at the University of Utah, where he's been since 2006. Prior to that, he was at Colorado State University, and if you look at his CV, you'll see that he's also had a number of very nice visiting professorships around the globe. He's an elected member of the American Association for the Advancement of Science. Among a wide range of scholarly articles on nanomaterials, there are several with a particular emphasis on nanotoxicity. The title of his talk this morning is Biological Interactions with Nanomaterials.
Thank you, Paul. So, I have no control up here for the launching this talk. All right. Okay. I decided to make the fatal error last night and go back and change my slides up based on what I heard yesterday. And we'll see how this works out. I wanted to express a certain message, and I hope that message comes through. I'm a pharmaceutical chemist. I directly inject nanoparticles into the human body for therapeutic purposes, as does my community. I think that over the course of 30 years in this field, that our community dealing with nanosystems has learned a lot, and I hope to try to convey that to you. So I focused this particular presentation on what happens to particles in the human body and to talk about in vivo consequences of nanosystems, because I think the pharmaceutical community has a lot to add to this discussion. So nanomedicine areas are a burgeoning field, a huge commercial interest. I think there's a lot of propulsion from the economic side, from the financial incentive side for a lot of companies to try to get products to market in the nano regime. In particular, there's nano drug delivery, which I'll talk about. But in addition, there's an enormous interest right now in new imaging and contrast reagents and what we call the see and treat modality. That is, see the lesion, treat the lesion. So if we can have a dual combination device, combination therapeutic, that can, in fact, image the lesion and then at the same time with the same vehicle deliver the drugs to that lesion, that's the dream scheme for the see and treat modality. We also have then interest in nanobiomaterials, materials that not necessarily therapeutic, but have adjunct properties for structural use, for regenerative medicine use, to support stem cell function, to guide regenerative medicine, for example. We also have nanopharmaceuticals that are not necessarily particulate, not necessarily materials, but in fact could be considered completely soluble. And that's a question about physics and chemistry, which we delved into philosophically yesterday, and I won't touch again today. The global market is, of course, an enormous incentive for this industry to move forward. By and large, most of the materials now in the marketplace are particulates. So the particulate market in general is a $1 billion market here in the next few years. Of that, pharmaceuticals and biomedical applications comprise a third, say maybe a fourth, 20% of that. And what we're seeing is that the cheap and easy things to get to market are, in fact, going there quickly, so that sunscreen protections, things we heard about yesterday, cosmetics, have the greatest near-term potential. But in fact, a lot of these are losing market share, not because they're not important, but because other things are coming up underneath, and the more mature development of longer-term goals is being realized. So these longer-term goals exist then in diagnostics and imaging technologies. A lot of these diagnostics, in fact, are point-of-care ex situ technologies that take body juices or biopsies or tissue samples and analyze them with nanoparticulates or nanoprobes ex situ, so they don't have to go into the body. So those are nearer term. Imaging technologies as well, for example, multiphoton, multispectroscopy technologies that are, again, ex situ outside the body but deal with body juices to analyze. What's going to happen is that the slowest to market are going to be those technologies that were discussed yesterday, in which a longer timeline, a longer FDA regulatory path, and a lot more money, $800 million in some cases, are going to delay some of these. And so the in vivo applications are, in fact, always, in my field, the slowest to market. So in drug delivery, we're faced with an enormous amount of engineered, clever chemistry. I see it every day as editor of 4 journals. I see it constantly on my desktop in my hotel last night, trying to edit people's creativity and not control it, but try to understand it. And so in my field, in my world every day, we deal with a plethora of fantastic architectures, of fantastic materials, of fantastic chemistries. And I know a lot of you haven't had chemistry since you're 16 or 18 years old, and I won't belabor these points, but the point of this graphic is to show you that we have then marvelous architectures that can capture drugs within a core shell structure, where you have a core that has the drug and a shell that protects the drug, or we have these micellar systems, which are quite now phase 3, advanced in phase 3 clinical trials against ovarian tumors in Japan. These polymeric micelles carry their payload here within this black core, and they have then these blue hydrophilic coronas that prevent unwanted interactions with biological components. An older standby has been the liposome. The liposome has an aqueous core that carries drug in its interior, surrounded by a lipid bilayer, much like the cell membrane, a cell membrane mimic, if you will. And then we can decorate that lipid bilayer with various things, the active targeting moiety, the magic bullet that can pull that targeted system to its intended site of action. We can have polymers that decorate the surface to prevent unwanted interactions. And there's various clever ways to decorate liposomes. This is a very mature form of drug delivery system. In fact, probably the only nano vehicle that is approved for use right now by the FDA is the Doxil product that carries an anti-cancer drug here and is decorated with polyethylene glycol chains and no antibodies. So just to destroy people's fantasies, there are very few directed active delivery systems where antibodies decorate the surfaces of particles and guide them magically to their targets. In fact, there are none that are FDA approved. We've heard in my field a lot about quantum dots. We heard yesterday about cadmium. I don't know where Maria is, but the cadmium is an instrument, a chemical alloy that's used to help produce these quantum effects and create interesting optical properties, but in fact it's highly toxic. So that these particles that are the fantastic quantum dots also do not have FDA approval for anything other than ex situ assays because of the toxicity associated with this particular combination of chemistry, which can never go into the body because of these problems with its intrinsic toxicity. And then highly branched, what we call hyperbranched or dendrimer systems, now trying to make it through clinical trials. There's Nanobio in Michigan that's trying to push this type of chemistry towards clinical trials in vaccine use and also in the delivery of iron for imaging and MRI systems. And so these are quite popular. There's many types of chemistries here. And then the standby would be paramagnetic iron oxide that's intrinsically magnetic, but as opposed to this branch structure is a solid black nanoparticle and can be decorated with various things to try to guide it to its system. So fantastic chemistries, but in fact, few have really made it into human use. So if we talk about medical product examples with functional nanotech components, there's drug delivery, diagnostics, analytical tools and instruments, for example, ex situ diagnostic and interrogation systems, medical material and implant biomaterial systems, and therapeutic systems. And I can't go through this whole thing, but in fact, I promised yesterday I would try to profile then how this is broken down across the ocean. So the next slide I put in talks about the fact that, in fact, in North America versus Europe, North America being in gray, Europe being in blue, that there is a difference in attitude, at least in how it's manifested as the products are developing. And this is data from Ernst & Young that I was able to get last year that talks about the fact that Europe is leading in analytical materials and instrumental methods. One reason might be is that they're most immediate to market. They don't invade the human body. And there's certain elegance in Europe in terms of making analytical instrumentation very cleanly and high performance. Europe is ahead of North America in the medical and materials implants game, but in fact it's beaten soundly by drug delivery. And this is exactly the point I tried to make in the Q&A session yesterday, is that the Americans and the North Americans in general are far ahead in the drug delivery aspects of this, while the Europeans are ahead in this. And this may be a cultural issue, may be an insurance issue, may be a reimbursement issue. There are many factors, and I don't want to place any judgment call on this. It's just the facts as I see them. All right, so if if I go to the central dogma here, drug delivery then, the deliberate administration of carefully engineered, carefully purified particles to the human body has provided us then a lot of problems, a lot of answers, and a lot of interesting experimental systems to teach us how these things behave, and I'll spend the rest of my talk trying to teach you how they behave in the human body. So we've got decades of research of particles interacting at various organismal levels, and the take-home message is that there are certain organs that really like to sequester nanoparticles, and it doesn't seem to matter right now what the chemistry is or what the physics are of these particles. There are certain organs that really like these particles, and other places where we can't get the particles to no matter what we try to do. And this has, I think, particular implications for how they're used in a therapeutic way, but it also has implications for how inadvertent exposure to nanoparticles might allow us to track those particles to known destination sites in the human body as well. So we concern ourselves in my field with the extracellular organ-level distribution of particles, and we call that biodistribution. If we inject particles, if we inhale particles for therapeutic purposes and we track them, the biodistribution is where they end up, and the pharmacokinetics is how fast they get there and how fast they leave. So two typical terms in my field are biodistribution, the end distribution of these particles, and the kinetics of distribution, the pharmacokinetics. So we can also talk about the barriers to preventing them from getting to their intended consequences. And circulation provides a barrier. We're going to see that blood components, the plasma fraction, the serum proteins, the platelet fraction, the cellular fraction, all have a profound interaction with injected particles that prevents them from getting to their destination. That is, injected particles rapidly partition into blood components. The blood components will facilitate their distribution, but oftentimes by binding them, also prevent them from getting to their endpoint destination. We have this curious system called the reticuloendothelial system, which comprises the liver, the spleen, the kidney, and in some cases the lungs and the bone marrow. The liver, the kidney, the spleen, the lungs, and the bone marrow. These four organs are the preferred destination for almost every particle that we inject or inhale deliberately into the human body. And so the reticuloendothelial system has been the bane of the pharmaceutical existence. Unless you want to target the kidney, unless you want to target the liver, trying to get it elsewhere, you have to bypass these organs. And in fact, they've had hundreds of millions of years of history of development that allows them to do their job precisely the way they're supposed to, protect the human host. And by doing so, they are then preventing a therapeutic from getting it to its endpoint destination. And I'll dwell on that point as we go along. And then at the cellular level, we have to consider, in fact, if we were magical and could get a payload of particles to a cell, that we have to get those particles then from the outside of the cell through the cell membrane into the cell. Typically, this is done by a transport system, by endocytosis or phagocytosis or pinocytosis, involving several different but related mechanisms of enveloping the particle into the cell membrane and vesicularizing it in a kind of processing garbage can. And the cell makes a decision point that it's going to destroy what it has taken in because it's, quote, dangerous, or it's going to allow that system then to escape into the cytoplasm and enact its action either in the nucleus, which is another membrane gate it has to get through, or at the mitochondrial level, another membrane gate it has to get through. So at the cellular level, we have at least three membrane barriers. This one, the garbage can barrier, and the nuclear or the mitochondrial barrier to get a nanoparticle. And so the fraction of particles that actually gets to a disease site that actually can traverse these three membranes successfully and find their intercellular target is vanishingly small. So that's why I have job security in my job, is to be able to improve that system so that oral dosage forms, for example, that comprise 86% of all medications that we take, an oral dosage form that could be nanoparticulate might end up with a fraction of 1% of its dose at its endpoint destination successfully. All right, so here's how my world is. If we look at old data, this is liposomal data. This is liposomes because, as I recall, the FDA has approved liposomes for use as a particulate injectable product, Doxil, for treating tumors. And if we look at the distribution then, the percent of the dose that arrives then, remember, at the reticuloendothelial system, liver, kidney, spleen, bone marrow, lung. Those five organs, greater than 90% of the dose of liposomes always, no matter what we do, no matter what we decorate it, no matter what the chemistry is, no matter what the physics are, they always go to this system. 10% in rodent models, which we know a lot about rodent models, 10% of the dose or less, if we're lucky, will go to the target solid tumor in a rodent. And if we then extrapolate that to a human case, we find, in fact, a fraction even less. So there's two take-home messages. One, no matter what we do, most of the dose goes here. A small fraction will, in fact, get to the dose site that we want, but in rodents, it's much more optimistic than we find in humans. So here's actual data. It's old data because this is an old field. I like radiolabeled data because it gives me precise radiolocation. So technetium-99 labeled liposomes, and we see, in fact, the black lines show the biodistribution, liver, spleen, bowel, kidney. And there's, of course, the fractions in all other places. But for this particular case, if we compare now the nanoparticulate technetium labeled system compared to just injecting the free labeled lipid, not particulated, into the system, we see a completely different distribution. So that the take-home message here is that the reticuloendothelial system is a daunting filtration system, it's functioning exactly how it's supposed to, but the nemesis for nanoparticle delivery. All right, so why is this? Can I get this out of here? Yeah. We have this theory in my field called the EPR effect. The EPR effect is called the Enhanced Permeation and Retention Effect, invented by Hiroshi Maeda in Japan in the 1980s. There's a nice review from him here I posted. And the idea is that the vasculature, the conduit by which then these particulates must distribute through the body, the vasculature is different in normal tissue versus diseased tissue. And in fact, we know that there are three types of vasculature anyway in the human body. There's continuous, where we have tight junctions, where the endothelial cells lining our blood vessels are in fact very tight, and they don't allow the escape of anything except those things that they want to by virtue of transport mechanisms, the receptor-based, very selective transport mechanisms. We also have fenestrated vasculature that we find in the bone marrow and in the liver. And this is then deliberately porosized vasculature that has pores because that's the way bone marrow and liver want it to be. So that fenestrated vasculature has pores in it. Then we have discontinuous vascular that characterizes certain types of organs, but also, interestingly, characterizes diseased and inflamed sites. So then in a diseased tissue, the vasculature becomes leaky. In an inflamed tissue, the vasculature becomes leaky. So in any disease or pathologic state, we have leaky vasculature. So the EPR effect states that normal vasculature is tight, diseased vasculature is loose, and if we can keep particles circulating long enough, that statistically, every time they pass a diseased site, a certain fraction will partition out through the leaky holes into the diseased state. So we see what we call passive targeting. If we can keep particles in solution long enough, and they circulate long enough, that certain fraction, a small fraction, but a measurable fraction, will partition out into the disease site. So nanoparticles work well for exploiting this EPR effect in diseased tissues, if, in fact, they're porous enough, and if we can keep the circulation lifetime of these particles long enough to statistically, every time they go around, a few will partition out. So I have a little movie here. See, I might jump out. And where is that? All right, and I'll show you. This is from a company that's now going bankrupt in Seattle. But they put together this video that I like. So this is a polymer that carries Taxol, anti-cancer drug. Injecting this polymer is going to be then a nanoparticulate of some form, and this drug is biodegradable. So here comes the drug down through a normal vasculature, and you'll notice that it's tight. All of the drug molecules shown here traverse the vasculature, but now here's an unhealthy vasculature with fenestrae or with pores, and we're seeing now that as the drug courses through, that certain amount of drug particles, not everyone, but a fraction will partition out through the fenestrae and find themselves now extravascularly in the tissue space. So this could be a tumor, this could be the meninges and meningitis, this could be in an infection site. So now if we zoom in on the polymer, so here's the nanoparticulate with the Taxol, and it's going to be now taken into the cell at the tissue site, and within the garbage can of the cell, the enzymes of the cell break the polymer up and free the drug, and now the Taxol becomes bioactive, and this is the story, destroys the tumor. So that's the cartoon version of all of this. And so if I can go back, F5, there we go. And so the idea is that we can exploit this for delivery of therapeutics. Now, the same thing would be operative for inadvertent particle exposure. So if you have occupational hazards or if you have environmental hazards, in fact, the same physiology would apply here. That is, disease sites or inflamed sites that might become inflamed because of the exposure of nanoparticles might then partition and selectively sequester particles in those endpoint destinations. All right, so the EPR effect, though, is not so simple. It's just that was the teaching slide for that idea. It's much more complicated. In an animal model where we exploited this then extensively over the years, we've cured hundreds of tumors by injecting nanoparticles containing cancer drugs to rats and mice that develop these lesions. And yet none of those therapies with nanoparticles have translated to humans, none. So clearly there's a translational issue in how we think about the EPR effect in going to a human model. And the short answer is that how cancers and disease develops in a human is much different physiologically and pathologically than what happens when we try to do a 2-week experiment in a mouse. By rapidly producing the condition, rapidly treating the condition, we don't get an extrapolation. The type of tumors are all not alike. None are homogeneous, so that we have heterogeneous types of tumors where we might very effectively treat a certain part of that tumor with the EPR effect, get the drug there, kill it. But there's other parts of that tumor that are just developing, don't have the same biology, don't have the same receptors, don't have the same fenestrae, don't have the same porosity, and therefore they don't treat well. And so we leave a fragment behind. The survivor tumor becomes metastatic and then ultimately reemerges as disease. There's issues. Once the particle gets out of the capillary, it just sits there in the extracellular matrix. It's outside the blood supply, but it's sitting in the tissue. Does it have legs? Can it just walk into the cell? I mean, what if it just engages into the extracellular matrix and just sits there? So this idea of what happens to particles once they leave the vasculature has not been answered. Delivery does not mean tissue penetration. It simply means delivery. The milk gets to your front door, but you've got to bring the milk in, put in the refrigerator. All right, some dependence on acute inflammation. So we know that when we engender acute animal models with acute disease and acute models that might or might not represent real human disease, then we can get this effect to work. But many diseases we're trying to treat in humans are not acute, they're chronic diseases. All right, and then I talked about the types of endothelium already. So we've rarely validated this EPR effect in humans to date or in large animal models, and it shows much smaller therapeutic effects in humans than in animals. Okay, enough on that. So here's one example I can show you for the EPR effect. This is penetrating the blood-brain barrier, the BBB. This is the holy grail for some of us of trying to get drugs into the brain. If we inject them in the arm intravenously, how do they get through the blood-brain barrier? So this was an interesting case in which commercial iron oxide nanoparticles, 10 nanometers, coated with a little slimy, slippery substance to keep them circulating so they won't coagulate out. These were injected into a human, and this is an MRI of a human brain, time-lapse, showing the development of images that show very nice lesion and structural physiological identification within the brain. And the idea is that this was very effective to inject these iron oxide nanoparticles intravenously and get them into the brain because this patient had meningitis. And so in meningitis, the swelling of the meninges membranes around the brain produced the inflammation that produced the discontinuous porous endothelial lining that allowed these particles to escape. So if they could circulate long enough, they could get into the brain and produce the imaging necessary. But there's rarely been other examples to show that this effect is real. All right, so we go then back to basics a little bit. Can we change this distribution? Can we change this behavior with various nanoparticles? So I'm going to walk you through a survey of the latest data I can find about how nanoparticles, when you inject them into a living organism, what happens to them. So the first thing to appreciate is what we heard about yesterday, but I like this slide, so I slipped this one in. The idea is that when we put everything from a 1 centimeter cube and now we cubify it into dust, and we make the dust into 1 nanometer cubes, that the surface area goes up, 10 million fold. So by simply taking a 1 centimeter cube and dustifying it to nanoparticles, we increase the surface area immensely. And we have then special surface and physical and chemical effects that we have to appreciate when we create nanodust and how that surface area interacts with physiological systems. So nanoparticles have specific surface areas that are very high, 800 square meters per gram. So the amount of nanoparticles you could hold as a teaspoon in the cup of your hand has the equivalence of 8 football fields of surface area. 1% of microparticles, large particles, only 1% of that microparticle's atoms actually occupy surface sites. But now when we dustify it and make it 1 nanometers, 10% of all the atoms in a 10 nanometer diameter metal particle reside on its surface. And when we go to 2 nanometers, 60% of all the atoms in that particle are on the surface. And since surfaces matter, we have to think about how that surface chemistry affects then the reactivity and the exposure to the human condition. All right, so let's go to the first case, 2010 paper from Prasad's group at SUNY Buffalo. Highly respected. He makes these silicon nanoparticles. This is a beautiful distribution if you're into this game. This is a fantastic distribution of silicon nanoparticles. If you look at the size distribution, it's very tight, around 20 nanometers. I like this data as an editor. I'm very happy about this kind of stuff. And he gives this then the zeta potential, which shows that these particles are negatively charged intrinsically because of their surface chemistry. And now he puts a near IR dye in them so he can use the dye to image where the particles go inside the mouse. And he shows 24 hours, 120 hours, 360 hours of circulation after injecting these particles into the mouse. Where do they go? The liver, the kidney, the spleen, and he's got some other, the skin and the stomach. And the reason he can detect them in the skin is because this is an ex situ method that's very sensitive to depth, as Maria talked about yesterday. If you're asleep, you wouldn't know that. But anyway, the skin has a certain sensitivity because of these ex situ methods of looking at them. So you always see particles in the skin. Yet if you inject the dye alone, no nanoparticle, you inject the dye alone, you see a totally different distribution. A lot in the intestine because the system gets cleared from the stomach, gets cleared from the bloodstream, goes to the kidney and the intestine. And then clearly, after 120 and 360 hours, it moves then away from these sites. So we have, the point here is that we have completely different biodistribution with the particles than we do with the dye alone. And that the RES system is healthy and active, and it doesn't matter that these particles are 20 nanometers. They go to the same places, exactly the same, as we saw with the large liposomes that are 200 nanometers, or with particles that are 1 micron. It doesn't matter. So here's then the images showing that we can label these particles, we can see them, and we see then the liver lights up, the spleen lights up, the lungs light up, the skin lights up. And then if we blow up the kidneys, we can see the kidneys blown up here. And then 2 hours post-injection, 24 hours post-injection, here's the liver showing how intense the liver sequesters these particles. And so if we look at the dye-detected nanoparticle distribution now from the dye, we see the liver, kidney, spleen, as we said. This guy was clever, and he put now an I-124 radio label on it. As if the dye wasn't good enough, we'll trace it with radio labels because they're very sensitive. And he shows then that the nanoparticle distribution with the radiometry shows, again, spleen, kidney, lungs, liver as the primary destination points for these particles. All right, state of the art. Next paper, 2009, UCSD work with Sangeeta Bhatia and Mike, I can't remember, guy in chemistry I know. Silicon nanoparticles, in vivo applications, Nature Materials paper. Same type of system that Prasad just reported. This time also in a mouse, injected, tail vein injection. And so what this data shows is that when you do the injection, of course, you can get the 2 solid tumor sites. They have ectopic solid tumors on the hind legs of both of these mouse. They inject it in the tail, let the particle circulate for a while, and what they see is that they get very high distribution in the liver and lungs here, so that we look at the kidney and the lungs. We see huge intensities of these particles lighting up in the kidneys and the lungs, and here we see the side of the mouse showing the kidneys and the lungs and the liver here in 3 side profiles. So yes, they can show that they can get some particles to the tumor site, but what they don't show, well, what they do show here, but they don't emphasize, is that most of the particles go elsewhere. So it's entirely consistent with what we know. It's not a bad result. For us, we're trying to get particles to the tumor sites. That's our job as pharmaceutical chemists. But in addition, we're trying to map then where these systems go. And it's clear to see from these data that we cannot avoid the RES system even with long circulating silicon nanoparticles that are luminescent silicon particles. We can map them with a radio imaging, we can map them with optical imaging, and we see the same effects. Here's another paper from 2007. This time it's an iron oxide nanoparticle. This is an iron oxide core decorated with a very hydrophilic polymer grafting that's done by Vinod Lapasitwar at Cleveland Clinic in Ohio, a highly respected nanoparticle expert. Again, the biodistribution profile over time shows immediate deposition in the liver and sustained deposition in the liver. Immediate deposition in the spleen and in the lung. So that these systems then go to the RES system. 95% of the injected nanoparticle dose of these nanoparticles that are now iron for imaging, decorated with this corona of hydrophilic polymer, go to the liver and the spleen at 6 hours. 90% in other studies that other people have shown. Again, hard to avoid the RES system. Here's now a whiz-bang polymer micelle. This is the system where we have Taxol and cisplatin loaded in the core, and we have this hydrophilic decorated corona, 20 nanometer particles, phase 3 trials in Japan for humans for persistent metastatic ovarian cancer and breast cancer. Very nice drug system. And so very nice chemistry. Here's the adriamycin drug conjugate. This is the anti-cancer drug conjugated to the polymer. The polymer folds up into this kind of particle decoration. Here's how it's shown in a stick form, where the hydrophilic corona guards then the hydrophobic core that contains the green drug particles. So if we look at this then in how it's distributed, the blood, you inject it in the bloodstream. Of course, the majority of the dose resides in the blood. There's an instant uptake by the tumor, which is good. But if you look at the y-axis here, the percent of dose that goes to the tumor, we have 6 or 7% that goes to the tumor, whereas if you look at the dosage that goes to the liver, 20% in the liver, 10% in the spleen, 8% in the heart. And we care about the heart because adriamycin is highly toxic to cardiovascular tissue, and so we care about how much goes to the heart. So if we do the time drive here and compile all those data, we see this huge load in the liver, the spleen, the kidney, the heart, and the blood, and a fraction in the tumor. So we get some where we want it to go, and the point is that if we could just push some from these other places where we don't want it to go, if we just get a fraction to go over the tumor, think about how efficacious this therapy would be. If it's already efficacious in humans at this level, think about how much it would be. So I'm trying to push my students all the time, not to look at the bad side, but to say, okay, just to skim off the top of the spleen, let's take some off the spleen and give it to the tumor and see if we can improve this therapy by targeting a little bit better. So here again, the micelle in a different system. This is a different polymer chemistry, 20 nanometer polymer micelles, this time with cisplatin instead of adriamycin, and the message is the same. This is in phase 3 trials in Japan, and we get reduced neurotoxicity that's intrinsic to the cisplatin drug, but the problem is that we get liver, kidney, spleen deposition, and some to the tumor. All right, that's state of the art. Let's move to some other things. Silver. So I found some silver data. There was so much emphasis on silver yesterday. Silver nanoparticles. And I can't vouch for this group. I read the paper, pulled the data last night. It was in a journal I don't read, Metallomics, 2010. So they have then taken these silver nanoparticles, they capped them with serum protein to make them serum compatible. They pre-cap them with serum protein, and they inject them into mice, and what we see is not surprising. The liver, the lung, the heart, the brain, the spleen, and you see that these particles all reside. This is the dosages that reside in those tissues after 24 hours injection. Most of them reside in the liver, the spleen, and the heart. All right, and this is the case when you don't inject particles, so this is the control. And so the point is, again, for me, it doesn't really matter. You can have metal. So we talked about metal. We talked about several different polymer flavors. We talked about iron oxide, and we talked about liposomes. 4 very diverse chemistries, same biofate. All right, and here's gelatin. So this is a collagen analog. It's a biomaterial. It's approved for use in humans. And this time they labeled it with radio labels, and only 4% of the dose of these gelatin nanoparticles gets to the site of action. Most of it resides then in the liver and the spleen. So I don't know what else to say to bring that message home. So what have we learned then? The classic biodistribution, that is the distribution to the RES pathway, seems to be, in my opinion, chemistry independent, size independent. 90% of any systemically administered colloidal drug ends up in the liver, spleen, lungs, and kidney, regardless of size, regardless of chemistry. When you talk about the physics, too, bring that in. Enhanced uptake in the liver, the spleen, and the lung and bone marrow is largely attributed then to the macrophages that line these tissues and the fenestrated type of vasculature that allows these particles to escape, because that's what the jobs of these organs is to filter and to purify and to protect the human host from these types of assaults. So anyway, the idea is that there's a difficult problem for us. So size does matter, though, in a way that the liver and the spleen clearance can filter a large variety of sizes. That seems to be very endemic. We can do macroparticles, nanoparticles, doesn't matter. But if we try then to put it, for example, to break the cellular barrier, endocytosis at the level of the cell, then what the liver does to sequester these particles of all sizes, in fact, is distinctly different than what the cell does at the level of the cell membrane, where endocytosis is, in fact, very size dependent. In fact, it's very nanoscale dependent. We like to see particles in the sub-micron range to get active cellular delivery. Cells don't actively endocytose particles in the super nanometer range. And the idea also is to pull particles out from the blood and have them partition into disease sites. We also then have a size dependence where smaller is better. 100 nanometer particles are much more effective in penetrating vasculature in disease sites. So size, in fact, does matter depending on what you want to do. And so for my students and for Pharmaceutics, this is a roadmap of how we try to tailor size to get to places where we want, even though we know that most of the dose is going to go to where we don't want. All right, and aggregation also affects in vivo clearance and in vivo toxicity. And there's a lot of work with carbon nanotubes that I tried to emphasize here because my other data did not emphasize that, with carbon nanotubes that shows how then carbon nanotube agglomerates, carbon nanotubes, how they aggregate, and how their history of working in serum, physiological, histological, biological environments, their aggregation state really changes how they behave in these conditions. And you have to be very careful when you review the literature about how the authors have reviewed how these conditions then aggregate, how they have treated that aggregation, how they've characterized it, and then how you know how the organism, whether it's a cell or whether it's a mouse, responds to that, because the agglomeration and the aggregation is very important to how they perform and behave. All right, so there have been many ways to try to then eliminate this aggregation behavior. And one of the common models, again, if we go back to the liposome, because this is FDA approved and this is involved in several products, the idea is to decorate then the surface of the liposome with hydrophilic polymer chains that act kind of like seaweed. And the seaweed is out there waving, and the seaweed is out there then preventing the adhesion of these particles to undesired sites. So we decorate the surface with these hydrophilic polymers, and one of the common ones is polyethylene glycol, PEG. And pegylation is a term that we use technically to describe the addition of these hydrophilic coronas around nanoparticles. And it's a very common pharmaceutical excipient. You'll find it in your shampoo. You'll find it in suppositories. You'll find it in aspirin. You'll find it in almost every pharmaceutical formulation. But in this particular case, the PEG is grafted in a decorative form to present itself as kind of a soluble seaweed that presents itself to prevent undesired biological responses. So this old data shows what happens when we take a naked liposome that doesn't have any PEG on it, and we inject it into a mouse, and we measure how much of the plasma concentration of the circulating plasma in the mouse contains liposomes. And you can see over time, this is hours now, over time, 100% of that dose is cleared within 10 hours. 100% of a liposome dose without PEG is cleared in 10 hours. Now we take the state-of-the-art method, and we decorate that surface of the liposome with PEG. This is the state-of-the-art, the FDA-approved product, Doxil. And what we see is that we can improve the half-life. The circulating half-life goes up to what? 12 hours. So the longest circulating liposome that we have circulates for less than a day. Because after 24 hours, 2 days. After 2 days, after 48 hours, almost 100% of that dose is cleared from the human body. So that's what we call a long circulating system. Inject it, 2 days later, it's gone. So the longest circulating therapeutic we have has a half-life of 12 hours and a circulating lifetime of 48 hours. That's what we call long circulation. So the idea is to present then the surface with these type of grafted chains, graphically shown here, to produce this effect going from this type of half-life to this type of half-life. And the idea in your head should be 48 hours is the longest we can get these particles to stay in the human body. All right, and there's a reason for that. Because when we inject particles into plasma, the plasma proteome at 70 milligrams per mil, the circulating juices in all of our bodies right now, 70 milligrams per mil of proteins contains 20,000 different proteins that span 10 to the 11th range in concentration, the most concentrated being serum albumin at 40 milligrams per mil, and the least concentrated being something that we probably haven't discovered yet, except that we know its genome from the Human Genome Project. So that's the problem, is that we have, against this daunting milieu where we don't know our enemy, except that we know the most abundant proteins, and many of those decorate the surfaces of nanoparticles and cause the liver, the spleen, and the kidney to recognize those particles and pull them out of circulation. So this opsonization event is daunting. And we have then a lot of different models for how proteins bind to nanoparticles. This protein corona method and how to interrogate it on nanoparticles is just coming to vogue on how to understand how proteins interact with nanoparticles. But we definitely have then a physical state in which aggregation and the dispersibility of particles and how individual nanoparticles agglomerate and aggregate in physiological milieu affects how they are recognized by human tissues and human organs. So, as I said yesterday in the question and answer session, nanoparticles are likely not lone actors in complex physiological systems. Most nanoparticles aggregate immediately upon contact with complex milieu. 70 milligrams per mil of protein is very daunting. Others bind to platelets, proteins, and red blood cells. So here's the cartoon from Mitre-Gauthry's paper that shows how nanoparticles are actually binding to red blood cells, and they're carried throughout the circulation. The circulating half-life of a red blood cell is 120 days. So when you see, somebody says, well, we've got particles that can circulate forever, the question is, are they really circulating because they're bound to red blood cells that are circulating? And eventually, how are red blood cells removed? By the spleen. So we find them eventually going to the spleen because they're stuck to the red blood cells. They can't escape the red blood cell fraction, and they find themselves in the spleen. So long circulating particles may be long circulating because they're attached, but because of that, they can't target. They're not going anywhere except hanging with the red blood cell as it goes around and around again. All right, so the last point is to talk about shape. Shape matters. So this is a nice paper in which people have made particles of different shapes and asked macrophages of the reticuloendothelial system that line the liver, that line the kidney and spleen, these macrophages to eat those particles and show us what they can do with different shapes. And what we see is that aspect ratio matters. And I bring this up because carbon nanotubes have a long aspect ratio, and it's very difficult to understand how macrophages could get their mouths or their heads or something around that. I was taught as a kid, never eat anything bigger than your head. And so the idea is that macrophages also have to deal with the idea of aspect ratio. And this point of this film is to show that macrophages that encounters a particle end on will rapidly invaginate and take this particle on end on, even though the particle is about the same size as the macrophage. And this map shows then the internalization velocity of particles as a function of their aspect ratio. And there are certain types of approaches and certain types of aspect ratios in which the macrophage cannot ingest the particle that's shown here. If a macrophage encounters the same particle but now side on, it cannot get its head around the side on particle. Same particle, end on versus side on. Interesting. And the corollary, here's the macrophage now trying to get a worm. So here's the purple worm. It's extended all the way through, and this macrophage is trying desperately to envelop this worm. And the idea is that if you talk about the number of particles that a cell can encounter, is that spheres are very easily ingested, but worms are not. And again, it's an aspect ratio issue, and the point of this thing is to show that the sphere is readily engaged, enveloped, and taken into the macrophage. And then at the bottom, the cylinder is a frustrated system. We call it frustrated phagocytosis. The particle cannot get in, even though the system is trying desperately to do its job. All right, so looking ahead, we need to distinguish differences then in reported effects in nanoparticle dosing. There's all kinds of people doing all kinds of crazy things in materials chemistry and the routes of administration, the contamination of these particles, the shape of the particles, and so we have to be very careful about how we compare apples to apples here and try to then understand how nanoparticles are in an inadvertent versus a deliberate then routing then taken into the human body. We need to validate dispersion and stability of these particles. I don't think nanoparticles last very long as individual particles in the human body. I think that we have to think about cooperative and aggregation effects a lot. We have to compare nanoparticles in vivo to standard reference materials. This was mentioned yesterday. We need to improve tools to detect how we know that nanomaterials are even in vivo. And that was mentioned yesterday also. We need to provide safety and efficacy testing guidelines to industry, because right now there is a huge confusion at the FDA about how these should be regulated. And we need to also show, and this is a classic problem for academics, that we can take all of these weird exotic chemistries that we make and we can scale them to GMP standards so that they actually become useful products. Because we'll never make a difference in human therapeutics unless we can take all that exotic chemistry and scale it under GMP facilities and under FDA QSR requirements. So nanotechnology has found some sound resonance with the political and business sectors. And here's the nano guitar, right? 2 micron scale bar here. It's about 50 nanometers. Each string is 50 nanometers, 100 atoms wide. But, but the biological harmonies are not yet so sweet. So anyway, I'll leave this for discussion, and I've just run over, and I'll try to take a question or two. Thank you for your attention.
David, that was very nice. Is that on? No.
Yeah, you're on.
Yeah, you're on. Okay. It was very nice indeed. I like particularly your EPR concept, and I'm wondering, we know from the air pollution field that people with chronic inflammatory diseases, diabetics, COPD, asthmatics, are specifically sensitive to ultrafine inhaled particles. Maybe it has something to do with the EPR concept.
Well, we do know that macrophages scavenge very actively particles in the deep lung, right? But we also know that certain types of particles can seemingly, and I like your validation on this, can seemingly penetrate the systemic to systemic circulation from the deep lung, from the alveolar space. Mm-hmm. Now, whether that's fantasy or not, I'd like some validation. So pulmonary delivery has a very low bioavailability, typically. We have less than 7% when we inhale drugs, less than 7% of them. So the idea is that somehow between the macrophage clearing that, and it can go to central circulation, carrying the drug, it can be the carrier of the particle, or it can clear centrally via the lung. So I do think that either we create an inflamed state that allows it to clear the lung, or we have other mechanisms endogenously that allow small particles to get through, or that the macrophages carry them into circulation.
Of course, I'm talking here about chronic conditions, chronic inhalation as well, yes? And so particles certainly in the inflamed lung get more easily across into the blood circulation. One other question was, when particles, you showed all these different particles pretty much behaving the same way. What has that to do, David, with the fact that they are, I don't know how quickly that goes, coated with plasma proteins? Maybe they are pretty much the same then once they get into contact with plasma, and that's why they behave the same way?
That's my feeling. Yeah, that's my personal feeling. And I think that there is a certain fraction of the community that believes exactly that. That opsonization by plasma proteins is such a problem, and we cannot select which proteins, no matter what surface chemistry we put on in there. We just simply get the same proteins on there. The liver says, oh, look, another particle, and just eats it. Right?
Thank you.
You're not on.
Is this on? Okay. Yeah. In my previous experiences in medical school, I remember two things. There are two sanctuaries in the human body from the immune system that I remember where you get some very interesting disease processes. One is the eyes, and the other is the testicles and ovaries. So I'm curious, have you seen any studies where they've tried to target these two sanctuaries?
Well, of course, yeah. The answer is no, but the anecdote for the eye is that the eye, particularly under certain pathologies, really is a classic example of the EPR effect. So macular degeneration, or any place where you have rapid angiogenesis, is the place where, in fact, these particles can be delivered. And I do know that Genentech and Roche are very interested in particulate delivery for the eye for particularly that reason.
It's also interesting because when the eye is physically damaged in someone that's otherwise healthy, the lens of the eye has never been exposed to the immune system of the person. And within a matter of 4 to 12 hours, the entire lens will be destroyed by the immune system. And so it's one of the aspects that we just don't understand.
Yeah, the lens is avascular too, correct? I mean, it's crystalline proteins and so forth. Yeah. The vitreous is an odd behavior. I guess I can't comment on the adrenal glands or the gonads, but I do know they're immune privileged. I just don't know how we target them.
Hello, over here. You discussed that particles are cleared from the bloodstream into the liver, et cetera, but I'm wondering about chemically inert particles, and that once they're in the liver, et cetera, what their lifetime is.
So we've been asked to look at gold, for example, because there's this fascination with Jennifer West technology at Rice University and these gold hollow nanoshells and delivering drugs with the see and treat modality, where you can use gold to see the image, and then you can heat the gold with ultrasonic or with radiation fields, and then release the drug by thermally stimulating the gold to do that. And we do see that we can gold plate. We saw a kidney image yesterday where somebody showed a gold-plated kidney. And I don't know what the clearance kinetics of those are, but we do see that much of the silver and the gold nanoparticles that go to the liver eventually go away. And the question is, how do they go away? And we don't know how they go away. Because if gold's really truly inert and it's not going to gold-4 cation, how is it getting to the kidney? We do know that the kidney can clear certain sizes of gold nanoparticles, but if we load the liver with particles that are clearly too large to clear by the kidney, where are they going? So it's a very good question. Very good question. I don't know.
Similarly with silica, things like that?
Silica, is, in fact, in my opinion, is not biosafe. Silica can dissolve. Inflammatory reactions can change pH wildly, so that the pH can go to pH 5.5 in tumors. So tumors that are hypoxic have acidic microenvironments, and also the endosomal vesicles within cells have a pH that's quite low, so we can dissolve silica. And I think that burden is well known. There is a very strange anomaly between amorphous silica and microcrystalline silica, where microcrystalline silica is highly toxic and amorphous silica is not. So the silica nanoparticles I showed, and those that are generally targeted by the community, are trying to make amorphous silica that dissolves much more readily than the microcrystalline form.
Very quick.
What's your opinion of what's going on with gadolinium in the MRI contrasts? This isn't exactly a nanoparticle, but in terms of the chronic disease questions, we're starting to see systemic sclerosis in people that get gadolinium contrast in MRIs.
Right. There's been some recent FDA warnings on gadolinium, and because there is a lot of interest in my community in trying to chelate gadolinium and control, I think it's a kinetics issue. If we can see the lesion and then quickly get it to the kidney and excrete it quickly, I think that we will get around a lot of the toxicity issues. So the idea is to create this window of opportunity where we inject gadolinium in a carrier, and there's enormous interest in my community in microparticle and targeted carriers for gadolinium that get it to the lesion quickly, get a high enough dose to get great contrast, and then within a couple hours it's gone. So it's the analog to a very short half-life radio therapeutic. But this is tricky. This is tricky because we know that most of that dose we're going to inject is going to go to the liver, and then we have to figure out how to flush the liver and flush the kidney and get it out before we see the kidney side effects and toxicity.
Okay, thank you very much.
Yeah.